There are corpses here. A burial took place that no one saw, and still, the mystery is closely guarded by the Canadian Rocky Mountains. The bodies have lain silent for nearly 500 million years. Only now is the evidence being unearthed, and the ghosts released at the fossil fields of the Burgess Shale.
You'll be taken on a short journey to the remains of a prehistoric community. Just as you come to appreciate the unique preservation and diversity of life forms here, you will learn of their destruction. Their extinction resulted in significant gene pool changes. There are 5 other factors that contribute to gene pool change over time. If these changes are significant and selected for by the environment, a new species could arise.
I'm kind of curious to know what we're going to see at the shale tomorrow.
What? And spoil this surprise?
I will share one thing with you, though. The fossils of the Burgess Shale are so incredible that they've been called everything from weird wonders to ghosts.
So what exactly do these ghosts look like?
Well, some have 5 eyes on the top of their heads. Others, headless. But the weirdest one of all is the one that has the spiny limbs on its backside and tentacles dangling from its underside.
Sounds too weird to me.
Well, tomorrow you'll just have to see it to believe it. I'll see you all in the morning.
Give me a break. Burgess ghosts?
Ooh, it's starting to give me the creeps. Sarah, why don't we head back to the tent?
Okay, but who's gonna put out the fire?
I'll put out the fire. I'm not afraid of the dark. Neither am I. Let's go find some water. Mary, there's a bucket over here somewhere. You can go first. Fine, stay behind, let the ghost get you. Oh, man.
Good morning.
Great. So what was all that ruckus about last night?
Must have been the Burgess ghosts.
Oh, you were visited by ghosts, were you? Any idea how old they might be?
I think they're about 1 billion years old.
About half that. About 550 million years, actually. The Burgess Shale fossils were thought to have been laid down during the Paleozoic era. Are you all familiar with the eras of the geological timescale?
Not really.
I remember some of them.
Do you think if I did a quick sketch here, you might be able to help me fill it in?
Sure.
Great.
If this column represents time in millions of years, and we start with 0 and go down to 3,500 million years, and on the other side, we have the name of the eras. Do you think you could fill in the names for me, Binez?
Well, the first era is the Cenozoic. I'm not too sure about the second one, but the third is the Paleozoic. And the fourth one is the Precambrian era.
Good.
I'm still not too sure about the second one.
Can anybody give her a hand?
Isn't it the Mesozoic?
Excellent. Could you fill that in, Binez? Great.
Okay, so how do geologists determine when one era ends and another one begins?
The boundaries between the eras marks major transitions between the life forms that have been found during those eras. Within each era, there are subdivisions called periods. It was during the Cambrian period that the Burgess Shale
Beds were thought to be deposited.
So where do humans fit into all of this?
Can I borrow that walking staff for a second, Cam?
Certainly.
If we can assume that this staff is about 1 meter long,
The base of the staff would represent the beginning of life, and the tip would represent present-day life. Then, if I take this dime and place it at the tip,
The thickness of the dime represents how long humans have lived on this planet.
And speaking of time, shouldn't we get moving before we turn into fossils ourselves?
That's a good idea.
The deposition of the Burgess Shale fossils is believed to have occurred during the Cambrian period, about 515 million years ago. In 1909, the Burgess Shale fossils were accidentally discovered by Charles Walcott when traveling through the pass on horseback. There are two quarries at the Burgess Shale, Walcott and Raymond. They are situated at about 2,300 meters elevation on Fossil Ridge. Fossil Ridge runs from Wapta Mountain to Mount Field in Yoho National Park. Across the valley is Mount Burgess. It's from this that the World Heritage Site got its name.
What makes the Burgess Shale so important?
The Burgess Shale, what you have is jellyfish-like forms, worm-like forms. You find the appendages of mineralized animals, but the appendages are preserved. So that what you see here is a much better idea of what the life was like at that particular time, half a billion years ago. It's also important because it shows us what the possibilities of life were, that it's evident from the range of animals which we get here that virtually every animal group which is alive today is represented here, and some others which we can't identify or classify very well, which may belong to extinct groups or may belong to groups who are so different from their descendants today that we can't recognize them very well. And that, of course, also, if they are extinct animals, it's telling us that more animals have become extinct since this time than we had previously realized. So it tells us a lot about the origin of life, and it tells us about evolution since then, particularly the importance of mass extinction. To the extent that if you follow the proposal of Stephen Jay Gould in One Wonderful Life, where he talks about contingency, that you could add contingency to mutation producing variation and natural selection, you now would add contingency or mass extinction as a third major factor in the process of evolution. So it covers it all. It covers origins, it covers extinctions, and it covers the process of evolution itself. And if you want to see one place in the world where you can actually see the evidence for this, this is the place to come.
Can you talk to us a little bit about the Cambrian explosion that led to all this variation?
Well, the Cambrian explosion, of course, is the name we give to the event when most animal groups appeared right at the beginning of the Cambrian period. There were some metazoans around just beforehand, the so-called Ediacaran fauna, but they mostly include jellyfishes, sea pens, just a few soft-bodied forms, not much variety at all. They were widespread, and then for some reason which we don't know, there was a trigger event which sort of caused this evolutionary explosion, and all these animal groups appeared. Now, of course, this was only possible because all the genetic variation necessary to allow this to happen had been built into the populations of unicelled animals, which had, of course, existed for up to 3 billion years beforehand. So you had 3 billion years of evolutionary development, variation developing slowly by mutation in these single-celled organisms. And when you had the event where single-celled organisms started to get together to form metazoans, multi-celled animals, all that variation was still in their genetic makeup, and so the possibilities were almost limitless.
You were talking to us earlier about the Anomalocaris. Could you explain a little bit more about that?
The claws of Anomalocaris were first described from Mount Stephen over my shoulder here in 1891. The first specimen was collected here in 1886. Now, at that time, I thought Anomalocaris was the body of a shrimp, and it had the name Anomalocaris because the spines on the side, which were thought to be appendages, weren't jointed. And all arthropods, because arthropods means jointed foot or jointed limb, so this was an unjointed limb-bearing arthropod. So in that sense, it was an anomaly or Anomalocaris, which means an unusual crab. Now, when Walcott started excavating in here, he got some specimens which he referred to a genus called Legania. He thought actually it was a holothurian, a sea cucumber, which had the mouth of this particular creature. But he didn't actually describe this animal itself. It wasn't until the Geological Survey of Canada, where he opened this quarry and the quarry higher up called Raymond's Quarry in 1966 and 67, that they got the first almost complete specimen. And in here we've suddenly discovered that the body of Anomalocaris was actually the claws of this animal, and that the jaws, the radiating jaws, which were well, I had a model here which shows what they were like. The radiating jaws of Anomalocaris, which Walcott thought was a jellyfish because it has fourfold symmetry, that this was in place as jaws. So we had the claws, which were thought to be a shrimp body, the jaws here, which were thought to be a jellyfish, is now his mouth, and then, of course, we had the rest of the animal itself. Now, when we reopened the quarry up there in 1991, 92, 93, we got an even more, a first really complete specimen, which showed the tail, which hadn't been seen before, and also showed that the head, which in the earlier specimen with the claws were retracted behind it. In this case, we have a specimen where the claws were drawn forward, and it was evident that the head was a lot more flexible than we thought. In fact, the whole head, eyes, jaws, claws, would all lunge forward to grab hold of whatever it was going to eat. So this reconstruction here, which is actually fairly accurate, is a bit different to the one that was produced in 1985.
The fossil animal, now known as Anomalocaris, was at one time considered to be separate fossilized organisms. The mouth was identified as an ancestral jellyfish, and the limbs as shrimp. Today, it is recognized as the largest predator that existed in the Cambrian seas.
It was classified as a World Heritage Site by UNESCO in 1981, before the Rocky Mountains World Heritage Site was created in 1985, and that now encompasses the Burgess Shale World Heritage Site. It's still the most special area for fossils in Yoho. It's the place where they're preserved their very best, and that's why Dr. Collins is still there. He finds animals that are so well preserved. You can see all the soft body parts, which is unusual for fossils. And they're still finding new animals there, so it's a really neat area.
So how come the Burgess Shale specimens were so well preserved?
Some scientists believe that there was very little decay here. Why? Well, one theory states that an avalanche of mud and silt rapidly moved into the marine basin where the organisms were living. The small creatures were swirled into disorienting clouds, some carried away, while others were buried when the mud compacted into the shale with each additional layer of sedimentation. So why is it important that there was so little available oxygen in this area?
Well, oxygen helps the decaying process. Without it, the decomposers can't survive.
Right. So with the decay process being so slow, the Cambrian community became very well preserved. You know, it is amazing that these fossils didn't get destroyed during the formation of the Rocky Mountains.
Look at the detail I'm getting just from making a rubbing of this fossil.
Yeah, that's nice, but mine is real art.
I can't get over the variety of specimens that they've collected just from this one site.
Yeah, me too. Miss Metcalf was right about the weird shapes and body plans. Hi, Mr. Fong, what are you up to?
Oh, hi, Sarah. We're just putting together a fossil here that we found. Do you want to give me a hand?
Sure.
This is just the third segment. Can you just put it underneath the microscope there?
Yeah, sure.
So then what we're doing, you have a microscope attached to a camera, which you're then going to capture the image into the computer.
Okay. Is that...
Yeah, just move that up a bit
Okay.
and then focus it a little. Okay, that's perfect.
All right.
So we just captured it into the computer here. And then once the image is in the computer, we can just add the piece we just digitized to the two pieces we already had.
All right.
Just copy it.
Why exactly are you piecing them all together like this?
Because we found this fossil in 3 separate pieces, and we digitized these 3 pieces separately, and now I'm piecing them together in the computer so that it looks like one fossil.
Okay.
And there we go.
Okay, well, this is a lot better than my sketch. Do you think I might be able to get a printout of this?
Yeah,
for my
that's
journal?
no problem. We'll just
Okay, thanks
print
a lot.
it right here.
Great.
And there we go.
Thanks a lot.
You're welcome.
The Hardy-Weinberg principle states that if the 5 conditions for equilibrium are met, then the allele frequencies in a population's gene pool remain the same from generation to generation. The Hardy-Weinberg equations P plus Q equals 1 and P squared plus 2PQ plus Q squared equals 1 can be used to analyze allele frequencies in a population over time.
I've just finished entering the data into this computer simulation.
We're sampling 1,000 fossils from 5 different strata of shale for the frequency of a homozygous recessive allele for spike-shelled organisms.
In the first strata, we found that 3 out of 1,000 fossils had a spiked shell. This means that the value for Q squared is equal to 3 divided by 1,000, or 0.003. This makes the value for Q the square root of 0.003, which is 0.055.
This means the value of P is 1 minus Q, which is 1 minus 0.055, and that equals 0.945. Okay, what if in the second strata, 100,000 years later, 37 out of 1,000 fossils showed the homozygous recessive trait for spiked shells? Then later in the third, 412, in the fourth, 480, and finally in the fifth, 448. Now how would this affect our graph?
Well, the graph shows us that the recessive allele increased up to the third strata and then remained constant. In other words, from the third strata to the fifth strata, the two genotypes remain the same.
So what might have caused the changes in the gene pool?
Well, there could be several things. Maybe those organisms that had the dominant allele of the round shells were wiped out by their predators.
So if the allele for spike-shelled organisms was a mutation, then it appears to have a selective advantage over the round-shelled organisms.
Or maybe a spike-shelled organism just drifted in from another region.
Well, whatever the reasons for the changes in allele frequency, we can see that the conditions for the Hardy-Weinberg principle were not met, and that the population is evolving.
The natural history of the world is not only found in the rocks, but is on display for tourists.
Weren't the fossils at the Burgess Shale so amazing? I couldn't believe it.
I love doing those rubbings. That was really neat.
Yeah.
Okay, what's the relationship between these prehistoric populations and populations today? Well, all populations, past and present, are influenced by two competing factors. There's the tendency to remain stable and the tendency towards variation. When a population is stable, what principle is being demonstrated?
The Hardy-Weinberg principle.
But those conditions usually don't exist in the natural population.
Was this the case with the Burgess Shale? This community was every bit as rich and dynamic as those that followed.
Well, the habitat must have become unstable, and as a result caused a change in the diversity of the community.
So why continue to study these and organisms from other fossil beds?
Well, to try to find similarities between populations that lived then and those that have lived since then. Once we find the similarities, maybe we will be able to better understand how the variations came about.
And what can be said about variation within populations?
Well, variations could cause changes in the gene frequencies in a population. If these changes occur generation after generation, perhaps a new species could evolve.
Wow, I don't think I could have explained that better myself. Now, you've all learned through your research that when scientists discuss the mechanisms for evolution, they don't speak of them in terms of the 5 Hardy-Weinberg conditions. They refer to them as random genetic drift, gene flow, mutation, non-random mating, and natural selection, with natural selection resulting in differential reproduction. These factors are different from the 5 conditions in that they show non-equilibrium in a population. Could you each take a minute to share your research with us?
Well, I'll start. I had a look at random genetic drift. Small isolated groups of individuals may have gene pools that drift away from the original population. Do you have a coin?
Yeah. What do you need it for?
For a short demo. Tails. Okay, if I flipped it 1,000 times, what would you expect the outcome to be?
Approximately tails 500 times and heads 500 times.
Right. Now if I flipped it only 10 times and the outcome was 7 heads and 3 tails, would you be surprised?
No, because you've only used a small sample.
Exactly. Small populations are similar to small sample sizes because the smaller the gene pool, the greater chance of random change in gene frequencies or variation from the expected outcome. Disasters such as earthquakes, fires, floods, and avalanches such as that of the Burgess Shale may cause a population to reduce significantly. Would the surviving population still be representative of the whole original population?
Not likely, but in the end it could lead to the development of a new species.
Right. Another example of random genetic drift is when a few individuals colonize an isolated island, lake, or habitat that is new to that species. Remember Charles Darwin's famous study of the finches in the Galapagos Islands? He figured that these birds strayed from the mainland of South America to the remote islands of the Galapagos as seen on the map. He believed that inherited variations within each population and the process of natural selection eventually led to the development of a new species.
The evolutionary aspects of gene flow or migration are interesting. Gene flow is the gain or loss of alleles in a population after individuals have migrated into or out of the area. The Hardy-Weinberg principle assumes no migration. If gene flow between two neighboring populations is extensive, and one gene pool is significantly larger than the other, then after several generations of mating, a new gene pool will be formed. Usually, it will resemble the larger population. Let's take human populations, for example. We're all traveling more and more around the globe. As we move around, gene flow may affect and alter gene frequencies within populations. For instance, the frequencies of the 4 main blood types in various races in humans. Within each race, certain genotypes appear to be more frequent. For example, 95% of Cherokee people have type O blood, whereas only 25% of Chinese people have type O blood. If any two of the populations shown on this chart were to interbreed, the frequencies of blood genotypes in future generations would most likely shift towards those of the larger population. Gene flow ends up reducing variation between two populations, and in fact counteracts the effects of genetic drift and natural selection.
Another agent of change in populations is mutation. Dr. Collins made reference to variation that was caused through mutation when speaking about change over time. On its own, mutation has very little effect on the allelic frequency in gene pools of large populations. However, when mutations are combined with natural selection and genetic drift, the effects are more obvious. After all, mutation is the major source of genetic variation. Keep in mind mutations are random, and there is a slight chance that they will be favored in the new population. I want to focus on mutation in bacteria. For bacteria and other microorganisms that reproduce rapidly, mutation is an incredible source of genetic variation. When bacteria reproduce asexually, they can divide as often as once every 20 minutes. At that rate, in just over 10 hours, one cell can give rise to 1 billion cells. If a bacterium should mutate and become resistant to an antibiotic, there could be millions of resistant cells in just a few hours.
We've all heard the expression love. It makes the world go round, but it also stirs up the gene pool. I want to talk about how non-random mating can lead to changes in the gene pool. The Hardy-Weinberg principle assumes that mating is random, but in actual fact, among animals, there tends to be many forms of non-random mating. Some organisms have harems where only a few males fertilize all the females. A contest usually occurs with some loud sounds, flashy colors, and threatening gestures. Other male organisms select mates similar to them. Humans usually choose someone who has similar tastes, lifestyle, race, social status, and athletic ability. Finally, there's sexual selection, where mate selection is the prerogative of the female. In male peacocks, they display bright plumage and females evaluate them and choose their mate. The most extreme case of non-random mating is self-pollinating plant species, for example corn, because they are selecting only themselves to mate with. This is restricting variation. So you see, non-random mating is yet another factor that can upset the equilibrium in a gene pool.
Yes, that's right. Those were some pretty impressive presentations that you've all made, but we're not quite finished yet. There's still one evolutionary process that needs some explanation. Cam, can you tell me what that one might be?
Natural selection. That's the way that more fit individuals pass on their genes.
More fit doesn't mean more strength or muscles. That's such a myth. It's closer to meaning better adapted.
Yes, that's a much better way of putting it than as. For instance, some individuals are more successful at producing higher numbers of offspring that live to sexual maturity. Therefore, the frequency of their genes will increase in the gene pool. If the environment favors these individuals, then they have been selected for. As environments change, natural selection will occur again over many generations until the favored genotype is maintained in the population. Let's take a minute and summarize some of the things that you've discovered. Sarah?
Mutation and genetic variation are the raw materials for gene pool change.
How does mutation affect different organisms then?
Mutations have less of an effect on organisms that reproduce sexually, because in most sexually producing organisms, the mutation would have to be dominant or occur in both parental alleles. But as you have seen in asexually reproducing organisms, such as bacteria, mutation is an incredible source of variation.
Good. Somebody else?
Yeah. Small populations that have become isolated from larger populations as a result of some natural event can be selected for. If the population can adapt to its environment and successfully pass on its genes for many generations, this could eventually lead to the development of a whole new species as well.
Could the isolated population still breed with the original one?
No, not if it has become a new species.
Good. Cam?
Movement of individuals into or out of a population could change the gene frequencies in the original gene pool.
Could you explain that a little further?
Let's say a lethal virus didn't affect people with A-type blood. If there were an outbreak of this virus on a remote island, all the people with B and O-type blood would either die or migrate away from the island. Thus, the gene frequency of the A-type blood would increase due to gene flow.
That's an interesting example. Blair, do you think you could comment on non-random mating?
I call it mate selection. Finding a mate can be quite the process.
The concept of a harem is quite interesting.
So is the female's prerogative to select the male of her choice.
I think you have the gist of this conversation. Let's move on. Now, taking all of the things that we've just discussed, if there's any change that occurs that could result in...
Evolution of a population.
Which could lead to...
The development of a new species.
Yeah, you've got it. There are different mechanisms by which new species may form. There are geographic mechanisms and there are genetic mechanisms for speciation. Today, biologists are debating whether the development of a new species occurs gradually over time or in quick spurts. Two theories that have received a great deal of attention are gradualism and punctuated equilibrium. In gradualism, speciation is a slow and gradual process. It is driven by the accumulation of many small genetic differences among populations. Changes in the body structures of organisms within these populations occur continuously over time. Large-scale evolutionary change is the result of selection among individuals, with the better adapted individuals leaving more offspring than the less adapted ones. In punctuated equilibrium, speciation is a geologically rapid event which is driven by fewer genetic differences among populations. Changes in body structures of organisms within these populations remain the same for long periods of time. Large-scale evolutionary change is the result of selection among primarily species and also some individuals. Species selection may in fact be the result of random chance. As we pack up, you might want to take a minute to review some of the topics that we've covered. Random genetic drift, gene flow, mutation, non-random mating, and natural selection. These are the factors that significantly influence population gene pools. Speciation is the result of change brought about by any one or any combination of these factors. Don't forget our visit to the Burgess Shale. It's shown us a bit about the past and has made us think about the present. What will the mysteries here and in other fossil beds unravel? Will our interpretation support gradualism or punctuated equilibrium? Or will there eventually be a combination of the two theories? We may never know all of the answers, but in the meantime, we've got some great ghost stories.